Hello everyone, welcome to C Academy YouTube channel. My name is Veleleni Nkosi. In today's lesson, we look at the process of the protein synthesis. By the end of this lesson, we will be able to see the difference between the transcription, which is part of the protein synthesis, and the DNA replication. Without any further ado, let's get into it. What you see on your screen is a lesson structure. So this lesson structure is based on most frequent asked questions in a protein synthesis. So I will read this so that you will be able to see what to expect in this lesson. So number 1, define the protein synthesis. Number 2, the importance of the protein synthesis. Number 3, describe all the processes of the protein synthesis, including transcriptions and the translations. And then number 4, which is the last one, tabulate the difference between transcriptions and the DNA replications. So this is the lesson structure of this video. So we will be focusing on this. I will explain as much as I can. So number 1, define the protein synthesis. So protein synthesis is the process by which cells build proteins in a body. So when cells build a protein, that process is called the protein synthesis. And protein synthesis, it involves 2 main stages. Stage number 1 is transcription, and stage number 2 is translation. So to explain those stages, during transcription, which is the first stage, the DNA sequence of a gene is copied into the molecule called messenger RNA or mRNA. So we have talked about mRNA in a previous video. So what happened in a transcription? An information is taken from the DNA and then by the mRNA molecule. So it takes place in the nucleus. And then in the translation stage, the mRNA is used as a template to assemble a specific sequence of amino acid into the protein. So translation takes place in the cytoplasm. So mRNA will move out of the nucleus, then used as a template. Then we will have another tRNA, then it's gonna come and build amino acid using the mRNA from the nucleus. So I will show all the processes as time goes, which is part of our lesson today. Number 2, the importance of a protein synthesis. So why do protein synthesis is necessary in our body? So the importance of a protein synthesis is to produce proteins in the body. So our body needs protein. So a protein is needed in our body for the reason why our body needs a protein for building and maintaining tissues. So tissues like a hair, for a hair to grow, there must be a protein that is produced in the cell. So we need that protein to grow a hair or our skin or many things. We use protein. And then many enzymes in our body are protein. So enzymes is like a catalyst. A catalyst is something that is speed up chemical reaction. So in our body, there's a lot of chemical reactions that are taking place. So for them to speed up, we need enzymes. So, and enzymes are proteins. And antibodies are protein. Antibodies are like bacteria fight. This works like antibiotics drugs. So they are fighting viruses and bacteria in our body. So it's like it's part of our immune system. So it's also a protein. So we need something that is fighting things like bacteria and viruses. Hormones like insulin and growth hormones are proteins. If you remember insulin, we study about insulin in grade 11 when we do animal nutrition. We know insulin is one of the hormones that regulate sugar in our body. So we need the sugar to not to be higher in our body, which is the functions of an insulin. And a growth hormone, this is the hormone that makes us grow. So we need protein so that a growth hormone can be secreted in our body, then we will be able to grow. So this is the reason why the protein synthesis is very important. So go into number 3. Number 3, describe all the processes of a protein synthesis. So we've got transcription, we've got translation. Transcription takes place in the nucleus of a cell. So to explain, this is the transcription. I got this info from a memo or a question paper, one of the question papers guideline, which was written in November 2019, paper 2. So this is the information. So I will read this information and then show you some animation of what happened so that it will be clear. Like transcription starts at the double helix DNA unwind. So if you remember the structure of a DNA, this is the structure of a DNA. So transcription takes place like only a part of a DNA, like from, let's say, gene 2. We've got gene 2, and then protein synthesis will take place only on this gene 2. Then from gene 1, nothing is happening. Gene 3, nothing is happening. It will take place only on the gene 2. So this is the difference between transcription and the DNA replication. DNA replication takes place in the whole of a DNA, while transcription takes place only in a part of a DNA. So they say that the double helix DNA unwind. So here we've got the double helix. What happened is the double helix will unwind. So it's no longer a twisting. If you can see, it's like it's no longer twisting around. It's just straight. And then number 2, weak hydrogen bond break or 2-strand unzip. So if you can see here, we've got like the strand are zipped right here at the nitrogenous bases. So for it, if it to happen, this will unzip. If you can see here now, they are unzipped, they are almost far apart. Then the next thing, to form 2 separate strands. If you can see here, we've got a strand here and then we've got a strand here at that particular gene. So like it's a gene 2 in our situation. This is a gene 2. We've got a strand here and then we've got a strand here. And then what happened next? One strand is used as a template to form mRNA, to form messenger RNA. So like here we will use only one strand. So let's use this underneath strand. This one will be unused. We will use this one. And then using free RNA nucleotides from the nucleotide. So in the nucleus, we've got free RNA nucleotide. If you can see, we've got, this is nucleotide, this is cytosine, this is uracil, this is guanine, and this is adenine. So these are free. They are just floating inside the nucleus. So we will use them to complete this template here. So like here, we've got thymine. So thymine, we know that it pairs with adenine. So we will take adenine, then adenine goes to thymine, and then again we've got adenine. Adenine will pair with uracil. We know that RNA do not have thymine, we have uracil. So A will bond with U, and then we've got cytosine. Cytosine bond with or pairs with guanine. Then we will go with guanine, and here we've got guanine, and then again we've got guanine. Guanine pairs with cytosine. So we need C. We need C here. And then here we've got thymine. Thymine pairs with adenine. And then here we've got cytosine. Cytosine pairs with guanine. And then we've got guanine. Guanine pairs with cytosine. And then you've got thymine. Thymine pairs with adenine. So here now we have built mRNA. This is the mRNA, is a messenger RNA taking information from the DNA. Then what next? mRNA is complementary to DNA. Yes, because if you can see here, this mRNA has completed the DNA. So now we've got a strand of a mRNA. From then, this mRNA will move out of the nucleus to the cytoplasm for translation. Then that is where we will use these templates to form or to produce proteins. So from there, we take this mRNA and then we move out of the nucleus via nucleopore. Then it's going out of the nucleus to the cytoplasm. Then the next step is a translation. Translation takes place in a cytoplasm. So the process of translation, this one I got from the exam guideline. Each tRNA carries a specific amino acid. When the anticodon on a tRNA matches the codon on a mRNA, then tRNA bring the required amino acid to the ribosome. So I will show you the process. Here we've got the mRNA moving out of the nucleus. Then mRNA will move out of the nucleus and then link itself on the ribosome. And then this is mRNA, and then 3 adjacent nucleotides of a mRNA, we call them codons, while 3 nucleotides of a tRNA, this is the tRNA, we call them anticodon. So ribosome will read this codon and then pair them with anticodon. Like here we've got A, U, G. So what the ribosome will need U, because U pairs with A, and then A, which is pairs with U, and then G, C, which is paired with G. So this tRNA here will fit here because A pairs with uracil. Uracil pairs with adenine, and then guanine pairs with cytosine. And then this tRNA has amino acid. This is the amino acid. So, and then after it puts here, then this ribosome will try to move to this direction. Sorry. Move to this direction and pairs another codon. Here we've got C, we've got A, we've got G. So C pairs with G, A pairs with U, and then G pairs with C. So we've got G, U, C. So this tRNA will pass here. And then after it passes here, this tRNA will leave the amino acid here. So let me ungroup. Sorry. Then this tRNA will leave and move and leave this amino acid here. So to look for another amino acid. And this amino acid, this one, will bond with this one. What happened is this one, let me ungroup here, then they will bond. Like there's going to be a bond in between. This one will come here, and then again also this tRNA will leave, then go to look for another amino acid in the cell, and then another tRNA will fill this space, and then they will also leave amino acids. So at the end we will end up having more amino acids with the information from the DNA. So we will have more amino acids. So these amino acids, if they are not yet made a protein, we call them polypeptides. They are not yet a protein. Like a protein that has a minimum number of amino acid is 50. So some of the protein contains more than 300 amino acid. But if it's not yet made a protein that is required, we call them polypeptide. And then the bond that is bonding this amino acid, we call it peptide bond. And then these things, when they are together, if they are not yet made a protein, we call them polypeptide. If they made a protein, we'll call them by the name of this protein. So this is how translation works. This is how the translation works. The information is from the DNA inside the nucleus. Then mRNA will move out of the nucleus with the information from the DNA, and then tRNA will come and then bond with the codons using anticodons, and they have to pair, they have to match. So if they are matching, then it matches, then leave the amino acids, and then another one will come and then match, it also leave the amino acid. So the sequence of putting these amino acids is from the DNA. So this is how the translation works. And then going further, we have like tubulate the difference between transcription and the DNA replication. So here's the difference between DNA replication and the transcription. So DNA replication, two DNA strands are used as a template. So in DNA we use 2 strands, and then in translation only one DNA strand is used as a template. So you saw that we only used one template, and a whole DNA is unwound in DNA replication. But in translation, only a part of a DNA is unwound, or a gene is unwound, and then free DNA nucleotides join DNA template. Free RNA nucleotide join the DNA template. So we've got free RNA nucleotide and we have free DNA nucleotide. In DNA replication, we use free DNA nucleotide because free in DNA nucleotide, they do not have uracil, and then these ones, they do not have thymine. So you must know the difference. Like here, they say adenine pairs with thymine. In the translation, adenine pairs with uracil. So this is the difference. This is from the question paper that was written in November 2022. So I got it from the marking guideline. So I think if you can download this paper, you will find the difference. So this is all for today. If you find this video very useful, please subscribe to our channel so that our channel can grow. Thank you very much. Anyone who's studying, I said good luck with your studies. Thank you.